So recording has now started for Chapter 3b of the course of Human Nutrition, where we will mainly talk about digestion processes in the GI tract. You can see an overview here on the slide, which actually gives you first the endogenous enzymatic digestion, which is mainly the digestion that takes place with enzymes in the upper part of the GI tract. So we're talking about the mouth, the stomach, and the small intestine. And we will mainly discuss that for three different parts. So the first ones are the proteins, the second one, the carbohydrates, and the third ones are the lipids. I will say a few words about the regulation, but not really in detail because it's a quite complex process. And then usually after the break, we will also discuss the microbial digestion, which is mainly focused on dietary fiber, which is not digested in the small intestine, as well as proteins. We will not discuss lipids there. There may be a small fraction of lipids that go to the colon, but generally speaking, the amount is quite small. And the most important substrates for the microorganisms in the colon are the dietary fiber and the proteins. So we will mainly focus on these ones. So what is digestion by using enzymes? We call that the endogenous hydrolytic digestion. The idea is that you're going to work with enzymes. There are several enzymes that are available in the GI tract. All of them can actually be used for breaking down different types of substrates. It could be proteins, it could be carbohydrates, like sugars, for example, and it could also be lipids, which can also be broken down in the small intestine mainly. The idea is that it is a progressive hydrolysis. So it actually means that several enzymes are working together, in which one enzyme first cuts, and then afterwards the substrate is transferred to another enzyme which further cuts it down, with the final end result, which are usually monomeric components like amino acids or monosaccharides or fatty acids. And these compounds are then ready to be absorbed, but in order to be absorbed, they have to be water soluble. The water solubility varies enormously. For lipids, this should normally be low. For carbohydrates and proteins, amino acids, it can be a little bit better. But in order to improve water solubility, we have a whole set of mechanisms available, which is mainly linked to emulsification in the case of lipids, so that the fat-soluble components actually become solubilized in a water environment. And this is required in order to get them absorbed and in order to get them transported through the human body. The whole process can be regulated. So it means that we have some tools available to modify a little bit the conditions in the GI tract. So we're mainly talking about the pH. You can play with this in the stomach and in the small intestine. The ionic strength, which is mainly linked to electrolytes, like for example chlorine or sodium, because sodium plays a quite important role in the absorption of different monomers, and the temperatures. But the temperature will not be changed here because we're talking about 37 degrees Celsius, which is the human body temperature. So in principle, you could change the activity as a result of temperature, but in our case, it will not be relevant. All these enzymes have to be activated. Activation means that you will secrete an inactive form of the enzyme, which is produced in the wall of the GI tract. And once it is released into the GI tract, the condition there will stimulate the conversion of the enzyme into an active enzyme. So for example, if we have pepsin, which is the enzyme that is present in the stomach, then we will release pepsinogen, which is a precursor of pepsin. It's produced by the wall of the stomach. You release it into the GI tract, and there the pH is so low that you have direct conversion of pepsinogen into pepsin, and then this enzyme becomes active. We will not activate it when it is still physically present in the GI tract wall, because if you do that, then the wall will be broken down. It is an enzyme that can break down proteins, The cell walls are fully composed of protein fractions, so when the pepsin will be active there, we can already have a destruction of the cellular wall, which we do not want to have. So in those conditions, most importantly, it is to start with an inactivated form, which becomes activated in the GI tract. So let's have a look to how the digestion process exactly works. And we will start first with the proteases. So the proteases are splicers of proteins, and they will actually break it down first to peptides and in the second part into the free amino acids. So here you can see an example on how it looks like. So this is the pathway on the left side, and on the right side you have different classes of enzymes. So the first one are the so-called endopeptidases, which will normally cut somewhere in the middle of the amino acid chain. And then we have the exopeptidases, which will cut on the outside and which each time will remove a monomeric component. The endopeptidases will cut at the level of the aromatic amino acids or at the level of the basic amino acids. And the examples are pepsin, released by the stomach, chymotrypsin, released by the pancreas, and the trypsin, also released by the pancreas. So let's start with these endopeptidases. So if you go to the left side, so you start with a protein, which we consume. In the mouth, not much is happening because there the pH is neutral, but we mainly have carbohydrate digestion there. So it immediately goes to the stomach. In the stomach, you have the release of acids. You have the release of pepsin, which is actually breaking down, as you can see here, near the aromatic amino acids. And so you have a cutting of the proteins into a whole mix of peptide fractions. And these polypeptide fractions are further broken down into dipeptide fractions by using trypsin and chymotrypsin that are actually further cutting down the peptides. And then in order to make very small fractions, which finally actually end up with the production of amino acids, we use the exopeptidases, whereby we have an amino exopeptidase, which is physically located near the glycocalyx. So it's here at the wall of the intestinal tract. And the second one are the carboxypeptidases, which are released from the pancreas. And so you just release them into the GI tract lumen, mainly in the intestine for carboxypeptidases. And it also means that it starts cutting from the carboxyl groups. The free compounds that you get are dipeptides, which may be absorbed, but it's with a question mark because I'm not sure that all the peptides will go in that way. But a few peptide fractions, especially the ones that are quite stable against hydrolysis, may normally be absorbed by specific dipeptide transporters. The free amino acids, which can also be released by the aminopeptidases, will then be absorbed into the absorptive cell, and there you have different types of transporters. There's one which is located near the basal amino acids, one for proline, there's one for the neutral amino acids, and one for the acid amino acids. So 4 different types of transporters, and all these transporting mechanisms are active. So it means that they require energy to absorb these compounds into the bloodstream. The energy is necessary because we use sodium in co-transport with the amino acid to bring the amino acid inside the cell and inside the bloodstream. The sodium outside the cell is always higher than inside the cell. We already discussed that last time. So sodium will diffuse into the cell, and in co-transport, it will take an amino acid with it. And this is the way by which the amino acids get inside the cell. In order to reinstore the original condition, we need to pump the sodium out of the cell in exchange, let's say, for potassium. And in order to do that, we need energy because sodium needs to go out against the gradient and potassium needs to come in against the gradient, and this requires energy. And it's a quite important step because about 25% of your daily energy requirement, which corresponds to about 500 kilocalories, is needed to maintain the sodium-potassium ATPase pump systems. So if you lack calories, automatically the sodium-potassium ATPase pumps will not work properly. And this also means that the absorption, in this case of amino acids, will not be 100% positive. So you get problems with absorption just to lack, in this case, of energy. Okay, so these are the proteases. So what is important for you to know is that you understand the mechanism. So the process as it is with the enzymes, with the transporters, with the sodium, with the conditions, sometimes acid pH, sometimes neutral pHs. So these types of things you need to know because you first describe it, but in the second phase, it could also be important to understand how you can modify the process. So how can we, for example, maximize protein breakdown or protein absorption? Or how can we minimize it? In case you, for example, would like to bring the proteins to the large bowel. In that case, we need to minimize the absorption. Or if you compare vegetable proteins with animal proteins, so both of them have different characteristics, different degrees of digestion and digestibilities. So you need to understand the process in order to understand why there is a difference between plant-based foods compared to animal-based foods. A practical example of using proteins actively to improve the absorption of nutrients is the example of amyloid fibrils. And I added a publication, I think, online on the Euphoria website where you can read it in case you would like to. So an amyloid fibril is visible here on the left side. So these are very small thread-forming structures which are stabilized by beta sheets. And they are produced, for example, due to heat treatment and prolonged heat treatment as a function of time. So in those situations, proteins will become folded, will interact, and will create a three-dimensional structure which looks a little bit like a spider web. So it's a little bit a thread-forming structure that can actually protect a couple of compounds when they are passing through the GI tract. So in the second line of experiments here, they actually made a complex between iron and the amyloid fibrils. So you have all types of dots here, which are a little bit linked to these fiber structures, and to which a compound, which in this case is iron, is bound. And it seems that when it is bound to these fiber-like structures, the iron is a little bit more stable for transport through the stomach wall and will more easily become available for absorption in the small intestine. So the last figures on the right are a little bit the characterization of the iron, that it is physically present within the fibrils, and during the transport through the stomach conditions, which is usually pepsin conditions and low pH, then you can see that actually the iron is physically still there, but it is attached to these amyloid fibrils. And this is the way by which it passes through the system. And in the end, it is actually brought to the small intestine where it could physically be absorbed. And the fibers are mainly stable at acid pH because you see here they are still physically there. In the case of adding the pepsin, of course, these are proteins, so to some extent they will be broken down. So not all the fiber structures will be maintained, and you get smaller fractions to which the iron will be bound, and then it goes to the small intestine for further absorption. So think a little bit, in case you know the principle of protein digestion, in case we give you a case study whereby you need to think about digestion, either to speed it up or to slow it down. Think a little bit how to do that, and also think about the potential application of this. So what could be the benefit of focusing on protein structures, digestibility, and absorption of nutrients? The second class of digestive systems is the carbohydrates. So in this case, we are actually working with sugars, and there are different types of enzymes available to break down sugars. So on the right side, you can see them. The first one are the amylases, whereby we have an alpha amylase and a beta amylase. The alpha amylase is present in the animals. The beta amylase is present in plant-based products. So for humans, we mainly are talking about alpha amylase, which could be physically present in the mouth, which is produced actually by saliva, as well as in the pancreas. So in case you eat food and you chew the food and you keep the food quite some time in the stomach, in the mouth, then the alpha amylase will be active. And you can actually already break down carbohydrates when they're physically present in the mouth. In case you don't really chew or you immediately swallow the product, so it stays a few seconds in the mouth, then of course you can expect that the alpha amylase will be active in the mouth. So in those conditions, the pancreas will produce alpha amylase, which needs to do the full job. Yeah, so it has to break down alpha amylase completely, or it has to use alpha amylase to break down the starch. So the alpha amylase is breaking down alpha 1,4 bonds that you probably already know from the previous lectures. So it breaks down alpha 1,4 bonds, whereby starch, Z-meal is called starch. In the course notes, everything is in English, so you can check it there further. This slide seems to be still in Dutch. So Z-meal means starch, and it will be broken down into, in this case, maltose. And maltose contains two monomers of glucose, which are linked together in an alpha 1,4 bond. So the alpha amylase will split the starch, you will get the maltose, and then the maltose can further be broken down by a maltase enzyme, which is actually present physically near the wall of the small intestine, and the end products of that are twice glucose. The glucose can only be absorbed by active transport. So it's exactly the same mechanism as what I told you before with the amino acids. So you need to pump in, well, not pump in, it just diffuses in of sodium that goes from extracellular compartments inside the cell. And in co-transport, you have the transportation of glucose coming in. And there's no energy required for that. You need the energy to pump out the sodium again and to bring the potassium in. And so that is why energy is required. The second example is saccharose. Saccharose or sucrose is a disaccharide which is corresponding to the table sugar that we have, the white table sugar, for example. So in this case, there is a specific enzyme which is called the saccharase, and this one is physically located near the wall of the GI tract. So you just break down, in this case, the bond between, in this case, fructose and glucose. Glucose is absorbed by active transport, and fructose is absorbed by diffusion. It's a sort of a facilitated diffusion, so it means it doesn't require any energy. There's a carrier transport that actually binds to the fructose and that physically absorbs fructose inside the cell. So the uptake mechanism of fructose is slightly different from the uptake mechanism of glucose. And that's quite relevant because in chapter 4, which is the next chapter, we will discuss the fate of fructose in the human body. And there you will see that if we take in a lot of fructose, for example, by using fructose syrups, which are quite used a lot in the US, Then the fructose can go to the liver, and in the liver, the fructose can be converted into lipids. So you get actually liver steatosis or liver fattening, which is something we do not want to have. Yeah, so fructose is a separate type of sugar with different characteristics compared to glucose. And then the last one, which I think, already discussed a couple of times, I'm not sure, is lactose. And this is a very important sugar because it's the milk sugar. So lactose is composed of galactose and glucose. And the bond between galactose and glucose is an beta 1,4 bond. Sorry, so not an alpha, but it's a beta bond. And a beta bond is not well digestible. So the result is that you need a specified enzyme to break down lactose, which in this case is lactase. It's also physically present near the wall of the intestine. So the end products are glucose, absorbed by active transport, and galactose, also absorbed by active transport. People who are lacking lactase, and they exist quite a lot in the world, are not capable of breaking down lactose. So in this case, the lactose will pass through the colon. In the colon, there are a lot of microorganisms there. They will break down the lactose. So in this case, especially if there's a lot of lactose there, you get formation of gases and you get diarrhea. So these people usually have cramps and they have quickly diarrhea because the lactose is not digestible by the classical enzymes. If you're not lactose intolerant, in those conditions, lactase will be physically active and you will produce the free monomers, which will be absorbed at the small intestine level. And then you don't get these extra symptoms. In the second semester, for people who are following the courses there, we will explain better what you can do against lactose intolerance. Yeah, so it's not true that if you're lactose intolerant that you cannot take lactose containing products. You can, but you have to take them in a very specific condition, which is mainly, let's say, a fermented condition or a condition whereby microorganisms are physically present to break down the enzymes for you. And if that is not possible, then the only option you have is to go for chemical or enzymatic hydrolysis at the level of the products. So some producers are already hydrolyzing lactose so that they have already the galactose and the glucose physically in the product before you start consuming them. That's also an option you have. So if we would look a little bit to the absorption of these monosaccharides, then we go just and have a check at the level of the intestine, whereby here you can see the absorptive cells. Here you can see the lumen of the gut. And on the other side, you can see the blood, where actually these compounds, which are amino acids or sugars, are physically being absorbed. And so they are not going to the lymphatic channels, but they are going to the blood because they are sufficiently water soluble for transport in the blood. So as we see here, so here you can see these transporters that I was talking about. Fructose is absorbed by facilitated diffusion, which does not require any energy. Glucose and different types of amino acids and galactose can actually be absorbed by the use of sodium. So there's a co-transport. Sodium is high outside the cell, is low inside the cell. So sodium is just diffusing inside the cell. And in co-transport, you take these other compounds with you, which is glucose or which is galactose or which are different types of amino acids. So they just come inside the cell. And then once they are in, they can go with a second transporter down to the bloodstream. This is the case for the fructose as well as for the glucose and the galactose. To reinstore the original situation, the sodium, which is now physically inside the cell, needs to go out. So the sodium is transported out in exchange for potassium, which comes in, and this is against the gradient. So that's why we need the energy, which is ATP transferred into ADP, and this is actually a quite energy consuming process. And so a lot of energy is required in order to achieve that. You may think by yourself, this is all quite a lot of details. Why do I have to know all these mechanisms? Well, a potential question that you might get is, tell me something about nutrition for athletes. You know, athletes, they need to have energy fast. They need to receive this energy from different sources, which is usually carbohydrate related sources. So a question could be, what type of food does an athlete needs to take, food or drinks, needs an athlete to take in order to have optimized metabolism during physical activity? Yeah, so it is actually a process whereby you need different types of compounds. You need to understand how are they broken down, how are they absorbed, what are the factors that are driving absorption. So if you know that, then you actually also understand what should be physically in the product in order to have maximized digestion and absorption. So you can think about it a little bit yourself. But what I want to say is we're talking about mechanisms, but I can translate the mechanisms into a practical case study where the mechanisms are needed to understand how to solve a problem. The last process that we are dealing with has to do with lipid digestion. And I must say that the lipid digestion is probably the most complex digestion process that we have, especially because lipids are not water soluble. And in order to get good absorption and digestion, you need to make them water soluble because the proteins are physically mainly present in the water compartments. So this means that the enzymes are there in the water compartment, while the substrate is a fat-soluble compartment. And these two don't mix. So you need to take specific measures to improve the digestibility of lipids. And the human body has adapted throughout time into a system which is extremely efficient. I would say that more than 95% of the lipids that we consume on a daily basis are absorbed in the small intestine. Now, only 5%, that's a bit the maximum, can reach the colon. All the rest is just absorbed in the small intestine, is metabolized, is stored without even reaching the colon. And that is due to the fact that we have a very efficient digestive process. In order to understand it, there are a couple of steps that you need to know. So the first thing is the enzymes are called lipase. And contrary to proteins and carbohydrates, lipases do not have a very specific substrate specificity. So they can be used for all types of fatty acids, long chain fatty acids, short chain fatty acids, saturated fatty acids, unsaturated, polyunsaturated fatty acids. All of them will be working with the same lipases. There is an exception for the phospholipases because the phosphate group, which can physically be attracted or attached to the diglyceride backbone, requires another type of enzyme. Otherwise, you get too much repulsive forces. So phospholipases are a little bit different from the actual lipases. The end product that you will produce is called a 2-monoglyceride. The 2-monoglyceride means that the fatty acid remaining on the triglyceride is at the 2 position. So at position number 1 and position number 3, so the outside of the triglyceride, there the fatty acid is released. And you produce, in this case, free fatty acids. which together with the two monoglycerides are forming the triglycerides. So once you get the formation of these two monoglycerides and free fatty acids, we need extra help to emulsify the product. Emulsification means that the lipids are solubilized in very small droplets inside a water matrix. So just imagine that you would take a glass of water and you would add a little bit of oil to the water. In those situations, there is no mixing, so the oil layer will be on the top while the water layer will be on the bottom. And in between, there is an interface whereby you have the interaction between the fat phase and the water phase. And technically speaking, if you keep it like that, this interface is normally the place where the enzyme can interact with the substrate. Of course, this will be a very limited digestion. It will be very slow because the interface is not that large. The enzyme needs to migrate to the interface, needs to attach, and then needs to break down the substrate. So it is a very slow process, which probably would mean that lipids will not be digested in time, and part of them will go to the colon. So in order to avoid that, we're going to make a so-called emulsion. An emulsion means that you have small droplets which are physically present in a water matrix. How can you do that? Well, you take your glass of water with on top your oil layer, and then you just start shaking. Shaking the glass as good as you can, put it back on the table, and then you will see that you get a diffuse layer inside the liquid. This means that all the different oil droplets are now solubilized in the water phase. And this creates a surface activity which is very high. So the interface between the lipids and between the proteins is extremely high in this case. Because you have all these small droplets, which is all one type of interface. Of course, the body of the system doesn't want to have that because it's not stable. If you just leave the glass back on the table and you wait for another 5 minutes, then you will see that the lipids will come to the top and the water layer will go to the bottom. Again, spontaneously. And so they just don't mix. So they just separate physically again because they want to have the smallest interface which gives them the smallest energy content of the system. And so the driving force is to get to the lowest energy content possible. So if we would just leave it like that, it would not be stable and it would separate again, which we don't want. So in order to stabilize the emulsion, we need bile acids. And these bile acids can actually interact at the interface between the water phase and the fat phase. And there they can stabilize the system. And they can keep the system for a longer time into the emulsified form. And this is ideal because the proteins, the enzymes in this case, are solubilized in the water phase, have maximum interaction with the lipids. And this is how actually lipids are broken down fast. We also need calcium, which is actually link up with the so-called colipase. And a colipase is a smaller enzyme that actually attaches to the interface, that pushes all the bile acids away because the bile acids are fully occupying the interface, which means that the real enzyme, the lipase, cannot interact with the substrate. So the colipase is there to first go inside the interface to push a little bit away the bile acids to make room for the lipase to follow, and then the lipase can actually interact easier with the substrate. So that's why we need a colipase and calcium in order to improve the activity of the lipase. And then the last part is the so-called enterohepatic cycle. And this cycle is actually responsible for recycling the bile acids. So the bile acids are released by the liver. They go to the gallbladder, where you have a sort of a concentration by removal of water. And then this concentrated stream of bile acids will go to the small intestine, will normally execute its work. So it will see that the interfaces become stabilized. And at the end of the small intestine, these bile acids are reabsorbed again. So they will come again into the body, go to the blood, and from the blood, they go back to the liver. So you get a recycling process, which we call enterohepatic cycling. So these are the key elements that you need to understand in order to understand lipid digestion. I will stop the recording for a few minutes so that you can answer or ask some questions. So recording has started again, and we have now explained all the parameters that are actually responsible for the lipid digestion process. So we can have a look now how the process looks like itself. So we have indeed these bile acid components that are different in structure. So we have different types of bile acids depending on the side chain, which is actually physically present here. Sometimes they bound, for example, to amino acids. And this means that the side chain structure can differ a little bit, but the principle always stays the same. So you have a background, which is actually water fat soluble, and then you have the hydroxyl groups, which are water soluble. And they reorientate next to the interfacial surface, whereby you can reduce the surface tension, which means that you will reduce the energy content of the system. And this is how you can keep an emulsion stable for a couple of hours if you want. So the average residence time in the small intestine is 4 hours. And by using the bile acids, you can stabilize the lipids strongly enough so that they can survive a couple of hours to be fully absorbed. If you don't have these bile acids, then the interfaces will be much smaller. You will not completely digest the process, and then not everything can be perfectly absorbed. These bile acids are quite aggressive. We have several model systems in our labs whereby we actually try to simulate lipid digestion. I'm saying try because it's not easy to make these emulsions on a lab scale and to incubate them with cells. This is quite a challenge. And one of the most important challenges we have is that the bile acids, if you add them to cellular cultures in very high dosing, so very high concentrations, then it seems that they are toxic for the cells. So the cells are actually disrupted, and they will penetrate inside the membranes. They will open up the membrane structure so that the cell actually lyses. And in order to avoid that, we need to make some adaptations in terms of concentrations, in terms of incubation times. So to simulate the model in the lab is still a challenge. With the water-soluble components, it's easy. We can manage quite well. With the fat-soluble components, we usually have problems in terms of stability. So here is the process as a whole. So I wanted deliberately to first explain to you all the different individual steps so that the figure for you will not become overloaded. But now by just looking at the figure, you will immediately understand how the lipid digestion process works. So here is the first part of the small intestine, which we call the duodenum. Then you have the intermediate part, which is called the jejunum. And then the last part of the small intestine is called the ileum. So here are the lipids. The lipids will enter in the duodenum. And we have the two types. So the first one are the phospholipids. The second one are the triacylglycerols. The first one contained phosphate, the second one doesn't. So you have the lipase. It's either a phospholipase or an ordinary lipase. It will cut away the lipids, and we already told you it's a two monoglyceride structure that is formed. So the first and the third lipid, so positioned on number 1 and position number 3, these are the ones that are cut away. And then you get what we call a monoglyceride, a monoacylglycerol or a monoglyceride. For the phospholipases, we cut away, in this case, the fatty acid at position number 2. So you see it's a little bit different from the classical lipases. And there you also get a structure that is a little bit surface active. The same as the two monoglycerides, because here is the glycerol, and the glycerol is partly released, partly not. So this compound will also be amphiphilic. So it will actually be partly solubilized in water, partly be solubilized in fats. And the same here. So part of it is soluble in water, the other part is soluble in fat. What we form then is a so-called micelle. So the micelle contains on the inside intact lipids. So intact triglycerides will go here to the center. And on the outside, you get the two monoglycerides, or these lysophospholipids, and that actually have released one lipid. They will be located on the outside, which already partially stabilizes the system. Yeah, so this is called a micelle. And these micellar structures will then further be stabilized by the use of the bile acids, which actually come from the liver, go to the gallbladder. From there, they are released in the small intestine, and they will actually bind to the micelle structure so that the micelle is fully stabilized and will maintain in that condition for a couple of hours. It can migrate here to the interface, and near the interface, you will release the contents of the micelle. So the two monoglycerides will go in, the free fatty acids will go in, not the micelle because that's too big, but the free fatty acids and the two monoglycerides will enter into the absorptive cell. This process does not require any energy, so it's a pure diffusion process because here the concentration is very high and inside the cell the concentration is very low. So you just absorb it inside the cell, and then you have a reformation of the triglycerides. So you can see with the ACAT enzyme, you just resynthesize the triglycerides, and these triglycerides are then packed within the so-called kilomicrons. And the kilomicron is a sort of a carrier, you could say, that actually is transported, in this case, to the lymphatic tissues, not to the blood, because it's still not very well water soluble. It contains quite a lot of lipids, so it will go to the lymphatic tissue. And these kilomicrons, in this case, will finally end up in the plasma, but this will be done once they pass the liver. So in other words, lipids that are absorbed from the body and the food will not go to the liver, they go immediately to the adipose tissue. So there's a bypass, and behind the liver they can go to the plasma, and from there they are transported directly to the adipose tissues. Why that is, you will see later, but you can already understand it. If all the lipids after absorption would go to the liver, then probably you get liver fattening, which we call liver steatosis, which is an important disease now in the westernized world. So in order to avoid that too much lipids are accumulating into the liver, we get a bypass, whereby the kilomicrons don't go to the liver, but just pass it. They go into the plasma, and from the plasma, they can go to the adipose tissue. And there they can be stored if necessary. So in this way, the mixed micelles are completely deplenished and depleted from all the lipids. And the only thing that is left in the end is just the bile acids. So the bile acids, in this case, will be absorbed in the last part of the small intestine, where you get absorption of the bile acids. They go directly back to the liver, and they can be reused. And that's why we call it enterohepatic cycling. So this is the way, the route to get them actually back into the liver. I hope the process is a little bit clear from you. It's a little bit complicated, but it's a very important process for you to understand. So I suggest that we take a break here of about 15 minutes, and then I will give you a first question, a problem-based learning question, which is based on this principle. So see you back in about 15 minutes. So welcome back again for the second part of the lecture on digestion. And we will start this second part with explaining the exercise, task 3A. So all the students that are present in groups A will have to solve this task. And the question is as follows. The hydrolytic enzymatic digestion of lipids in the small intestine has been explained in the lectures. Based on these principles, which opportunities are there to develop a so-called light product, which is a product on the basis of lipids that supplies less calories to the body? So the idea is that you go back to this mechanism here, that together with the members of your group, you look carefully to the mechanism and you try to identify these elements that can be modified or changed in such a way that the absorption of calories from lipids is reduced. So how can we do that? Which types of steps can we introduce so that we still eat lipids, but the lipids do not supply enough calories? That's the idea of a light product. So how would you do that and try to motivate your answer? If you look carefully, you will probably find several opportunities or several techniques. So the one that you are finally going to choose would have to be the one that will give you the least amount of side reactions. Because always if you do something different, according to this process, you might introduce secondary effects. So please think a little bit further which technique or which options, strategies would be the best in order to avoid side reactions and still meet the objectives of reducing caloric uptake. This is the task whereby I ask you to only use the course notes and of course the discussion with your members to be creative. And if necessary, you can look in the reference articles that are on Euphora. But I do not recommend you to go online because on the examination there is no online connection. Yeah. So if you really want to practice it, it's better to do it the way it would be on the examination. The answer should be maximum 10 lines. And that's more or less the volume of the answers that I expect on the real examination. So don't make it a very extensive discussion. 10 lines is enough, and upload it by the deadline, which is indicated in the program. Okay, so we can continue now with discussing a little bit the regulatory system. You can imagine that this is a very complex system because there are many hormones involved in trying to regulate GI tract digestion in humans. And I plotted 4 of them that you can see here, which is gastrin, typical stomach hormone, the CCK, which is cholecystokinin, pancreasemin, which is located and linked with the gallbladder and the pancreas, then the secretin, pancreas and stomach, and the GIP, which we already discussed in the previous lecture, gastric inhibitory peptide, with targets stomach and intestine. You see here the different action points, so the different conditions which finally lead to this hormone activity. And finally, you can also see here the stimulus, so the effect that is actually causing the hormone to be released. For example, for gastrin, we know that when the stomach is getting full, then you get a stretch in the stomach wall, and this is releasing gastrin. While for CCK, this will be fatty acids and amino acids that are appearing in the duodenum. This will give you the CCK release. The secretin will be released once there is food in the stomach and the intestine, And the GIP, once you have carbohydrates and fats physically located in the duodenum, then the GIP will be released, which actually will help you to stop the activity of the stomach. So the system is actually fully autonomous. It will work on itself. So the only way by which you can influence the system, so in other words, influence the release of the hormones, is mainly by influencing the geometrical condition of the stomach. So, for example, if it fills a little bit less quickly, then probably less quickly gastrin will be released. Or you can also modify the composition of the food, because the composition of the food that finally ends up in the intestine will also determine the type of hormone that is physically released from the GI tract. Yeah, so you can actually quite easily, by looking at the table, understand which techniques we have in hand to try and modify the hormonal release patterns. If we apply that a little bit to two different conditions, then you can already see the potential practical applications. So on the left side, you can see the effects that can finally lead to activity in the stomach. While on the right side, you can see the interaction between the stomach on the one hand and the other organs on the other hand, which is in this case the liver, the gallbladder, then the pancreas and the small intestine. And so they all interact with each other thanks to the use of these hormones. So for example, if we go to the left side, it's a small figure, but I hope everybody can more or less see this. So here you have the glands, which we already discussed in the last lecture. So we know that from the glands, there is a release of acids. And this acid release is actually stimulated by a whole set of factors. So the first one is by bringing food into the stomach, because then the stomach wall is stretched, and then all the glands here will come under pressure, and then you will more easily release the acids. That's the first stimulus we can have. The second one is by the use of specific types of food components. So, for example, if you wake up in the morning, you don't have time to eat, so the only thing you do is to drink a very strong cup of coffee. Then the caffeine in the coffee, once it enters in the empty stomach, will also be a stimulus for the release of acids from the stomach wall. So it means that you get more acids into the lumen of the stomach without any food there that can buffer it. So the final result is that you have, quite a strong impulse or strong effect here at the level of the stomach wall. So if we look to a second stimulus, which you probably also know, this has to do with strong peppers. So these very strong, small, red pepper-like structures, they're extremely strong, and they can also release, in this case, acids from the stomach wall. So especially if you eat spicy foods, or even if you take the peppers without the food, that's even stronger, then you get a very strong acid response. So which can actually act on the mucus layer in the stomach, and which can lead in the end to potential stomach ulcers. And then a final mechanism is also here coming from the brain and directly acting on the release of the acids. We call this a cephalic response. So for example, you haven't eaten, you pass next to the canteen or a kitchen and you smell the food, then these aroma components will actually already induce secretion of acids in the stomach. Think about the dog of Pavlov. It's a little bit a similar mechanism whereby the dog gets food and at the same time you ring a bell. And you do that a couple of times and then you only ring the bell, but you don't give the dog food. And then in this case, at the same time, you have release of acids because the dog already knows that food is coming. So he's preparing a little bit the digestive tract for the entering of the food. So a lot of mechanisms by which you can control acid release in the stomach. Here you can see the interaction between different organs. So in principle, when food is entering in the small intestine, and the stomach in this case becomes empty, then the stomach is not needed anymore. So in this case, you can actually look at the composition of the foods and you can actually give a feedback that goes back into the stomach in order to stop the normal activity of the stomach. And that's why we are releasing GIP once the stomach is empty, because at that moment you here have the food already physically present in the stomach, in the small intestine. You can give a feedback to the stomach to stop working. If the stomach is full, so it is a little bit stretched, then you release the gastrin, and the gastrin can physically activate the stomach activity. And the CCK, for example, it acts on all these organs, which can all be done by just measuring the content of the intestine. And this will actually help you by releasing CCK by the wall of the intestine, which can then act on the gallbladder. It can act on the pancreas to release all these juices so that you can have normal digestion in the small intestine. So think carefully when you are just looking at all these systems that you try to imagine what can happen in different conditions. For example, what happens when the stomach is empty? What happens if I give more lipids? What happens if I give more proteins? So how could that influence these regulation of these organs?